Welcome, everybody. It's a pleasure to have the opportunity to speak to you today. I teach a course for Yale undergrads called Immunity and Contagion, and several of my students said they're thrilled to be in the course this semester because of what is happening with COVID-19. I also do research on the immune system. So I've divided my talk into three parts. First, what are viruses? Second, immune response to viruses. And third, vaccines and their enormous impact on disease prevention. So just a little background to get started. Viruses are extremely small. So on the left, you see a typical eukaryotic cell. That's a cell that we have in our body. We have trillions of such cells. We have skin cells, kidney cells, blood cells, different varieties. They all have a membrane around them. They have a nucleus in which the genetic material is found. That's DNA for humans. And then they have different organelles and machinery out in the cytoplasm, such as mitochondria, which is involved in energy, and ribosomes, where proteins are translated. Then we have a bacteria, and you can see the relative size of the bacteria. The bacteria also has a wall around it, or a membrane, and it doesn't have a nucleus, but it has also genetic material, in this case, DNA. Then you can see the size of the virus. So the virus is much smaller, and there's two basic types of virus. They have inside genetic material, which is either DNA or RNA, and then they have a shell around them, which is made up of viral proteins, and some of them have a envelope which they take from the membrane of the cell that they're infecting. So you can see that they're very, very small. This image is of a cell that's infected on the right here. And you see these little projections from around the outside. We can magnify that, and it turns out those are viruses that are budding off the membrane. And you can see there's hundreds of them coming off this cell membrane. So what are the features of viruses? First of all, they can't exist on their own. They have to infect cells. Once they infect them, they take over the host cell machinery and multiply inside the host cell. And they start really growing exponentially. They then spread to new people before a person dies. Otherwise, they will not be able to be transmitted and continue to propagate themselves. Now, the coronaviruses were found in animals in the 1930s, and then were discovered in humans around 1965. And this I took from the talk that Dr. Landry gave to have us all on the same page, because some people did not attend that webinar. So two of them were described in 1965 as cold viruses, and additional two in 2004, and they are about 15% of the viruses that cause the common cold. But then in 2002, the severe acute respiratory syndrome coronavirus emerged in China, spread to different parts of the world, and fortunately, we were able to contain that, in part because people who contracted it generally got sick, and it was we were able to identify those people. That caused about, though, 10% deaths in people that contracted the SARS. Then in 2012 was the Middle East respiratory syndrome coronavirus that emerged in Saudi Arabia. That has about 35% mortality, but it doesn't spread well between people. And the people that have contracted it initially was from camels, because that is the animal that carries it. And then they didn't transmit it well except for very close relatives or healthcare workers. But it still remains a threat. And then in 2019, we had the novel coronavirus outbreak in Wuhan. One of the things about these viruses is that they are highly mutable. They exist in bats. There's hundreds of coronaviruses in bats. And in the case of SARS, in these wet markets, it looked like the bats transmitted it to a civet cat, which then was able to transmit it to humans, and then human-human transmission. And in the case of the MERS, it also is thought to have originated from bats that then went to camels. So I want to start by saying a few things about cold viruses and about seasonality, because that comes up. The rhinoviruses are responsible for about 50% of all virus detections, and they're also commonly found in children with asthmatic attacks that go to the hospital. Now, this curve was from a study in which they looked at respiratory viruses in hospitalized children and adults over different times of the year. So on the x-axis is the months, we see spring and fall, and on the y-axis is the number of cases. And in the front is flu, influenza A. That's one of the varieties of influenza. And you can see it peaks in the winter months and is much less the rest of the year. In contrast, the respiratory viruses, you can see fairly substantial numbers throughout the year, some peaking when children return to school. And this is similar to what happens in the community as well. We don't know with this COVID-19 what the seasonality is going to be, and that's a major question. The other thing about these cold virus infections, which is really so interesting, that we were able to really do this kind of study because of advances in our ability to detect viruses. So in this study, they looked at 25, by Byington, they looked at 26 families over a year and took nasal swabs from each member of the family and asked, do they have a rhinovirus? Can we detect it? And the number on the first row is children under 5 years old, and the dark shows those that were positive, and you can see about 50% were positive. And then as you get older, the numbers go down. But of the ones that were positive, only half of them actually had symptoms. The rest were asymptomatic. Asymptomatic people can still transmit the virus. And so we have this in coronavirus where we have asymptomatic carriers. So in these respiratory viruses, they initially go into, let's say, our nasal cavity. And just like we have skin that lines the outside of our body as a barrier, when we go inside, we have a layer of epithelial cells, and they're orange. They're shown to be orange up there on the image on the right. They're orange, and they're tightly packed together, and that helps create a barrier. And they have little cilia at the top that sort of tries to move things out, any particular matter. And also we have yellow cells called goblet cells that secrete mucus. And the mucus lines our epithelial airway, line our airways. And so if you've ever gotten sick, you know you make a lot more mucus and you can cough up phlegm. And that's all protective. So if we get the infection up at the upper respiratory tract, you see the virus is infecting the epithelial cells. We're making more virus. We see the symptoms of the common cold: a runny nose, nasal congestion, sore throat, headache. Now, if the virus goes down to the lower respiratory tract where we have the lungs, this can result in more severe illness, because the lungs are critical for gas exchange and our ability to breathe. And what happens when people become very ill with the coronavirus is that they develop severe respiratory distress syndrome, and they have real difficulty breathing. So let's go to immune response now to viruses. There are many types of immune responses to microbes and pathogens. As an analogy, if a fly enters your room or a raccoon, are you going to respond the same way? No, you're going to respond very differently. And that's the same with our immune system. We have to tailor responses depending on the type of organism. And so I'm just going to focus today on viruses. So the steps in immune response. First of all, we have to recognize a microbe is present, that it's actually entered our body. There's no point in making a response if we don't have a microbe. Then we have to activate the cells to make a response, the immune cells or other cells of the body that detect it and fight the infection. And then after it's cleared, we have to stop the response. So you can think of a car, you put your foot on the gas pedal to make it go, but then you have to stop at some point and you have to put your foot on the brake. And there's active mechanisms for stopping immune responses. And then you have to repair the damage. Because when you're mounting a response, cells are dying, a lot of things are happening, and you have to repair the damage. The immune system has two basic arms called the innate and the adaptive. The innate is in many, many organisms, including plants, insects, mammals. It's a very ancient system of immunity. And this is a fast response that occurs in hours, and it has the ability to recognize classes of microorganisms through these special receptors, which I will talk about. In vertebrates, adaptive immunity developed. And this immunity takes time, 5 to 7 days. It's very, very specific to the microbe. And also we generate memory cells. And this is very critical in helping us to be protected if we ever get exposed to that microbe again. And for long-lived species like us, that happens. So first, let me define a receptor. We have receptors on the outside of cells. You can see a cell here with the nucleus, and you see an image of a receptor. We have receptors for taste, for smell, for hormones, and for microbes. This first discovery of receptors for microbes, classes of microbes, was recognized by a Nobel Prize. It was such a fundamental discovery. Now, if the receptor binds to a target molecule, like it's called a ligand, if that binds to the receptor then will transmit a signal inside the cell, and the cell then will make a response. For microbes, there are about 60 different receptors that detect classes of microbes, and they recognize conserved structures on these microbes. So that makes it harder for the microbe to escape. So we have some examples of one set of them called the toll-like receptors, TLRs. Some see bacteria and parasites, number 1 and 2. Some see gram-positive bacteria or fungi, 2 and 6. And in circle, we have a receptor, TLR7, that detects single-stranded RNA, which is what coronavirus has. And this is a receptor that is in the inside a vesicle called an endosome. So the coronavirus, just to remind you, has that red single strand of RNA, which is recognized by that particular toll-7 receptor. And it also has an envelope around it. And then it has the spike proteins. These are viral proteins that stick out on the outside. And when a picture of the virus was first taken with an electron microscope, it looked like a crown. And so that's why it was called corona, for a crown. Now, when it first infects epithelial cells that line the airways, those cells on the left that I mentioned, it can also be detected by another receptor that's inside the cell. So on the right, if you look at the image of inside the cell, you have a coronavirus there with little spikes. And the spikes bind to a specific protein on the surface of certain cells, epithelial cells, called AC2. And once it binds that, it is able to infect those cells. If the cells don't have AC2, it can't infect. So it binds, and then the membrane kind of pinches in, and you get a little vesicle called the endosome with the virus inside. The virus particle then, the envelope around it, merges and fuses with the endosomal membrane, and the genetic material, the RNA of this virus, gets released. And there's a receptor called MDA5, which then binds this, sends signals into the nucleus, affects gene expression. So now the cells start producing two very important molecules called interferons and inflammatory cytokines. And I'm going to describe what those do. So on the left side of the picture, again, if we go back, these start within 2 hours. And once one cell is infected and it starts making these interferons, it can then, those interferons can then bind to neighboring cells to alert them, hey, I'm infected with a virus. You guys better get ready and start preparing to mount a response against this virus. So let me first say something about cytokines. Cytokines are how cells communicate with each other. There are hundreds of different ones. The interferons are one class of cytokines that then signal to other cells. So on the left, you can see an inducing stimulus. That would be the receptor that would detect the microbe. The cell would then make the cytokine, and the neighboring cell that would have a receptor for that cytokine would then bind it and get a signal. And when the neighboring epithelial cells bind the interferons, they turn on 300 genes that produce proteins that will interfere with every step in the virus life cycle, from infection to making multiple copies of itself and replicating, to budding, etc. The other class of cytokines are called inflammatory cytokines, and they induce inflammation. Now, probably many of you have experienced inflammation. For instance, you may have a sport injury, or you've had an infection, and what you get are swelling, redness, warmth, pain. The blood vessels start allowing the cells lining the blood vessels start opening up so that fluid and other cells can come into that area to potentially fight an infection and bring nutrients, etc. And this is a very important immune response, but it does lead to tissue damage, which has to be repaired. Now, another thing cytokines do is they communicate with the brain that make you feel sick. So you can ask, why do I have a fever? Well, the cytokines that are released also tell the brain, we have an infection. And fever, temperature, can actually help against certain microbes, actually, the increased temperature in the body. It can lead to altered sleep, lethargy, anorexia, you don't feel like eating, social withdrawal, depression. You won't necessarily get all of these, but these are some of the characteristics. Now, besides the cytokines, the interferons and the inflammatory cytokines, there are also cells that are part of the innate immune system. I'm just going to talk about a few key ones. One is the macrophage. So the macrophage is like a guard cell. It's located in tissues. So in the lung alveolar tissue, you have macrophages there. And they can also sense a virus, and they will secrete the interferons. And the same with the other cell here listed, the dendritic cell, both of them will. And this is an image of the macrophage. So macro means large, phage is from the Greek word phago, or eat. And in this picture, you see the macrophage is holding on to some little green bacteria, and it will actually take it inside of the cell, eat it, degrade it, and spit it out as a degraded substance. And so it kills the bacteria, and it can do the same thing for viruses. Just as a little aside, for those of you who have tattoos, it turns out you might wonder, when you inject the dye, why doesn't the dye just go away? Well, it turns out the macrophages eat the dye, but they can't digest it, so it just sits inside the macrophages, and they're long-lived cells, so the dye just stays there. Now, another cell I want to mention are the neutrophils. These are the foot soldiers. They're the most abundant, other than the red blood cell type in the blood. And when there's inflammation, the macrophage also sends out signals, cytokines, to recruit them to help fight infections. And they're short-lived cells, and when they die, they actually can lead to pus. So if you see pus, it's kind of white, those are dead neutrophils. And then we have tissue-resident lymphoid cells, which I'm not going to go into, but these cells also are working to fight in the infection. Now, this is our initial response, but vertebrates also have, those are organisms with a backbone, also have two cells that are very key, called T cells and B cells. And I'm going to be talking now about what those cells do. But first, I want to mention that in order for them to become activated, the dendritic cell, which is a sentinel, is a key cell type. So what happens is, if you have, let's say, an infection in your foot, a nail goes in, you get bacteria in there, the dendritic cell, which is that star-shaped cell, will take up the pathogenic material, and they will move to a lymph node through lymphatic vessels where your T and B cells are located. And they will initiate an help initiate immune response. So they're a sentinel. So the innate immune system is letting the adaptive immune system know we have a microbe here. So actually the lymphoid system is composed of lymphatic vessels, which are all throughout your body. And in yellow, you see the vessels, and then the little yellow circles that connect the vessels are lymph nodes. And you can see they're under your arm, they're around your neck, they're in your gut area, they're really throughout your body. And the T and B cells will circulate between the lymphatic vessels and the blood vessels, going through lymph nodes until they encounter the foreign antigen and this dendritic cell to alert them. There's a couple other cells can alert, but I'm focusing on the dendritic cells. Now, what happens? So the adaptive immune system has two components. The T cells are for cell immunity, and the T cells themselves fight the microbe. And the other is humoral immunity with the B cells, and these make antibodies, and I'll tell you what these are. So we have millions of B cells that exist in our body, each with a unique receptor for detecting a foreign antigen. And you can see that little Y shape on the B cell surface, and each of the four shown here have a different receptor. They're very specific for a particular antigen, the thing that's immunogenic. And in this case, we're showing a virus. When it gets triggered, the particular B cell then will begin to multiply and proliferate so that we have more of them. Because we have so many millions, we don't want to have so many of each one. So we only have maybe 100 of each one, and it will start multiplying. And some of the cells will develop into a plasma cell, which is making antibodies and secreting them, the Y-shaped antibodies. And these antibodies will have a number of different functions. The plasma cells are long-lived so that we can have small amounts of being antibodies to a particular microbe being made for years. The other thing is we have a long-lived memory B cell, and this cell can also be involved in mounting a rapid response if we get reinfected. So one of the functions of antibodies is neutralization. They block the ability of the coronavirus to infect cells. So here we see a schematic of the coronavirus where an antibody against the spike protein is binding, and it blocks the ability of the spike protein to bind to that ACE2 protein on the membrane, and therefore it cannot infect. So these are neutralizing antibodies, and many vaccines against viruses are inducing neutralizing antibodies to prevent viruses from infecting. Then we have the other cell-mediated response called T cells. So the T cells are called T because they mature in an organ called the thymus, which is right above your heart. And these killer T cells can develop against the pathogen. And what they do are kill virally infected cells. So I have a little schematic here. So we see an infected cell, and on the surface, there's a protein called class 1 MHC, and it carries little parts of viral proteins to the cell surface. And that's how the killer T cell knows that this cell is infected. So these MHC molecules are really important for bringing little parts of proteins up to the surface to let an immune cell know, hey, I'm infected. And then that cell will kill it. It releases toxic granules with toxic substances in them to kill the cell, and the cell will die. And then the killer T cell will move on to kill another cell. Now, some of the cells will become killer cells, but some will become memory cells, which are long-lived, because after an infection, we want to get rid of the killer cells. We don't need so many of them anymore. And this is a little schematic of a kinetics. So we see on the x-axis days, and on the y, we look first at the virus. The virus enters, it starts multiplying from 3 to 5 days, but then after 5 to 7 days, and even earlier, really, we start getting a T cell response, as well as actually an antibody response. And that hopefully then is controlling the virus. And then after the virus is gone, there's a contraction phase where these killer T cells will die. We don't need them anymore. But the memory cells that have developed will live for years. Now, what influences immune responses? First of all, there are external factors. Temperature, interestingly, here at Yale, they did a study on these respiratory viruses in the nose, and they found that when the temperature is cold in the nose, the immune system doesn't work as well. There's also data on pollutants and certain ones that affect immunity. Then there's internal factors. If people have lung disease, aging, our immune system declines with age, and just like a lot of other things decline, unfortunately. So we have that. Obesity is also a factor. Some people are immunosuppressed for various reasons. For instance, there's a treatment for people with multiple sclerosis. This is an autoimmune disease in which the body attacks myelin that lines the nerve conducting. And they want to suppress the immune response in this autoimmune disease, so they're on certain therapies. Well, that leaves them potentially vulnerable to certain types of microbes. And then interesting genetics. They did a very interesting study about children that were hospitalized for respiratory virus infections. Now, most children do get this by age 2, some type of virus. And these were children who were perfectly healthy, and this is the first time they'd ever been hospitalized for that kind of a problem. And they did a genome analysis, and they found that in 8% of the children, 8% of the children, they had an alteration in one of these receptors that would detect the class of microbes. So they were not detecting it that well or not signaling that well. So how does this information relate to vaccines? First, I want to say that one of the greatest medical advances has been the development of the smallpox vaccine and our ability to eradicate it. So last century, smallpox killed 300 million people. In the 1790s, Edward Jenner introduced vaccination with cowpox to prevent smallpox. And actually, he had a lot of resistance. And even when he died, it was not completely accepted. In 1960s, there was a global vaccination effort to eradicate it. And in 1977, it was eradicated. Now, because smallpox does not infect any other species, the virus cannot return to infect people. And that is not true, though, for viruses that live in other species, such as Ebola, coronavirus. But this is a really fantastic accomplishment. Now, what's in a vaccine? It's usually a form of a microbe that won't make you sick. They can be inactivated or weakened viruses or some component of the microbe, which is what the human papillomavirus vaccine is, a component. And they will induce memory cells and long-lived cells, these plasma cells, making antibodies. So why is that protective? Well, we have a little schematic here of the levels of effector T cells, the killer T cells, and antibody after a first infection over time. Now, if we get reinfected a second time, where it says second infection several years later, the response is more robust and faster. And that's because we have the memory T and B cells, which are rapidly activated, and there are many more of them very quickly. And then we also have these protective antibodies. Now, vaccines are important for a community. First of all, not everyone can be vaccinated. Some people have these immunodeficiencies. As I mentioned, children on chemotherapy are immune compromised. But if we have enough people that are vaccinated, the virus can't be transmitted to the unvaccinated people. And that's called herd immunity. Now, if a virus is highly infectious, such as measles, then a greater proportion of the population must be vaccinated. And in this, for measles, it's estimated that 95% of the population needs to be vaccinated. And the little image just shows you if no one's vaccinated on the left, then the disease can spread. If some of the population gets vaccinated, it spreads through some. But if most of the population is immunized, then it really contains the spread of the disease and the microbe. Now, vaccines do take time to develop. First of all, you create the vaccine and you hopefully test it in animals if that's possible, if there is an animal model. You then have to check for safety. That's the first phase of a clinical trial. Then the second phase, you do small scale efficacy. And the third phase is large scale efficacy. And then you have to set up for production and distribution. Now, you need people who are actually exposed to the virus in order to test efficacy. If nobody has the infection anymore, you can't test it. And you use controls and test cases to do this. So this is an important area of research, as well as research on viruses, on our immune system. And I put my plug in for the importance of research to keep us safe. So a couple take-home points. The innate immune system is our first line of defense. There are multiple mechanisms to sense invading viruses and to fight against them, such as interferon, inflammatory cytokines, and innate cells. The adaptive immune system, we have the creation of specific antibodies to neutralize viruses so they can invade cells, and we also can create killer T cells to kill virally infected cells. Interestingly, the presence of antibodies in the blood of people indicate that they were exposed to the virus or a vaccine previously. So if you want to know if asymptomatic people were ever exposed, you can look for antibodies in their blood. You won't find the DNA in your nasal swabs, which is what they're doing for assessing people who are currently ill, to look for the presence of the DNA, of the RNA of the virus. But you can do this to see if people have been previously infected. And vaccines lead to the development of these memory cells that produce a stronger and faster response. So thank you for listening. I'm happy to answer any questions.
Dr. Kavathas, thank you so much for taking the time out of your busy schedule to do this for us. That was an excellent presentation. I know it's a scary time for a lot of people, so I believe the more information that we have, the safer we'll wind up being. So thank you again for doing that. We do have plenty of questions, and please do submit them in the chat feature on GoToWebinar, and I will get started. I'm going in chronological order. First question, let me see. Who should be quarantined?
All right, so my first disclaimer is I am not an MD, but I would say this, right now, what they are doing is quarantine people with known contacts of infected people. And so that, I think, is a minimum of who should be quarantined.
Okay. What is the turnaround time for the COVID-19 test?
I think it's about a day. It's not going to take weeks. Our older tests did take days, but this can be done within a day by taking the nasal swab, amplifying the genetic material from the virus, and then there are tests to determine if viral sequences are present. And they can also sequence these viruses. And there's very interesting work going on at the Fred Hutch in Seattle, where they're looking at transmission of these viruses by sequencing them and looking at the mutations that they have and creating evolutionary relationships by looking at which mutations are present in which viruses and how they potentially evolved and moved.
Okay, thank you. If someone recovers from the virus infection, does that mean they no longer have the virus inside them and cannot infect other people?
Well, in order to infect other people, you have to have active virus being produced. And so if you don't have active virus being produced, you're not going to infect other people. And to my knowledge, these are not viruses that just live inside of you for long periods of time.
If someone is ill but not tested and could be a COVID community-acquired infection, how long are they to stay in isolation once recovered from fever and shortness of breath?
Yeah, that's a medical question that I don't think I can answer, unfortunately.
Fair.
But I have to say that the testing is very important. And I recently talked to a colleague of mine whose family is in Italy, whose mother has fever, cough, but she's not sick enough to go to the hospital. And it turns out they're not testing those people. They're really just testing people that are in the hospital. So the health system's kind of overwhelmed, but if we really want to know more about transmission and spreading, it would be useful to be able to test anybody who's symptomatic.
Absolutely. COVID-19 is active in current summer or warm weather locations, for example, Australia. Wouldn't that suggest that it would still be active during the North American summer?
I think we just have to wait and see. We really do.
Right. Are individuals that get rhinovirus-induced asthma suffering worse from the coronavirus, or is that data not yet available?
That data is definitely not available yet.
What is involved with getting tested for COVID-19? Is it a blood test?
It's usually a nasal swab.
Mm-hmm.
So they're looking for the presence of the viral genetic material in the nose.
Okay. Are there any particular points to emphasize for access and functional needs and homeless populations to remain safe?
I don't think I can really answer that question.
Sure about
Sorry.
That one. Sorry about that. Is it true that one of the complications of the COVID-19 virus is a cytokine storm? Please explain.
Yes, that is correct. So I talked about cytokines being made, correct? And it turns out if you make a hyper response and have a ton of cytokines, you can have a huge amount of inflammation. And I talked about the fluids coming into the tissue that's inflamed. And so if the lung has a great deal of inflammation, you get a lot of fluid in the lungs, and this can cause a lack of ability to breathe.
How effective are simple methods such as opening windows to increase the diffusion of fresh air in an enclosed space?
Again, I think that's a question that's outside my field of expertise. I could speculate, but
appropriate quarantine venues or locations versus not. In other words, why keeping people on a ship is not a good idea?
I don't want to comment on those things, unfortunately.
Fair enough. Yeah, we've just got so many questions here.
I know. I think anyway.
Yeah, we'll get to we'll Dr. Kavathas will answer to the best of her knowledge. So forgive us for only answering some of these. How many days before symptoms start showing would the testing show positive? How many days after symptoms subside would the test still show positive?
I don't know that answer, and I'm not sure people exactly know yet either, because we haven't done enough testing.
Right. Once someone recovers from COVID-19, will they be immune from getting COVID-19 again, similar to those who have received a vaccine for another virus like smallpox?
Yeah. Presumably, if there's no genetic alteration or mutations in the virus, you should have adequate immunity after you recovered, if you're a normal, healthy person.
Okay. Can any of the work done on SARS and MERS vaccine development expedite a COVID vaccine?
I would say yes. I would say yes. Unfortunately, they never really got to making the SARS vaccine. They started it, but the infection died out. But I think that they were working actively on that, and that potentially can also help.
Thank you. What would testing outside of the hospital environment involve? Home kits or kits prescribed over virtual mediums?
Oh, I'm not sure about that. Dr. Landry could easily answer that question because she is head of the virology lab at the Yale Hospital, and this is what she does. So if you send in your questions regarding this, we could forward them to her and get some answers that way.
Yeah, absolutely. So I have all of these questions that are being asked documented. So we're working on, like I said earlier, on the write-up of Dr. Landry's presentation, and we'll do our best to include all the answers to these questions. So thank you. Alrighty, and I think that is the last question that we'll cover for today. We very much appreciate all the attendance. And again, Dr. Kavathas, thank you so much for your time today. And if you guys would like to receive the recording of this webinar, please shoot me an email at Julie, Julie, at Veoci.com. And again, I'll be sharing the write-up of Dr. Landry's webinar as well once that's complete. Thank you again for participating today, and everyone have a great rest of your week. Take care.